Hello guys, this is Vineet and welcome to the Molecular Biology series, chapter number 5. So today I'll be discussing about the reassociation kinetics of nucleic acids. Okay. So before we get into the actual concepts, let us try to understand the basic terms of the main concept, that is contour length, C-value paradox. Okay, what are the basic things about the denaturation and the renaturation of the DNA. Okay. So now contour length is also defined as the total length of the genomic DNA in a cell. Okay, whatever the nuclear genome is there inside a cell, that is called as a contour length. Okay, so we also have the formula for to calculate the contour length. Okay, so for the formula for calculating the contour length is, you have to multiply the number of base pairs present in the nuclear genome with the distance between the monomer, or the nucleotide bases. Okay, what is the distance between the two bases? Okay, so that is multiplied with the total number of base pairs that is present in your nuclear genome. Okay, so if we look at this human genome, okay, so human genome approximately contains the 6 into 10 to the power of 9 base pairs. Okay, and the distance per monomer is what? 0.338 nanometers. Okay, so if you multiply that, the length would be approximately 2.32 meters. Okay, so this is what the contour length is, and we also measure the contour length by using the atomic force microscope. Okay, so they already have a scale in that microscope, or the application. Okay, so by visualizing the DNA or the plasmid DNA or any segment of the human genome segment. Okay, so they measure the length, contour length, and they calculate the number of base pairs, or the actual distance between the monomers. Okay, so this is what about the contour length. Contour length is what? The total length of the genomic DNA, that is calculated by multiplying the total number of base pairs with the distance between the monomers, or distance between the bases. Okay, so the distance between the bases for the diploid cell of a diploid human cell is 0.338 nanometers. Okay, in the nuclear genome, the distance between the base pairs is 0.338 nanometers. Okay, so this is what the contour length is. And then we have the C-value and C-value paradox. Okay, so if C-value refers, it refers to the amount of DNA that is present in the haploid nucleus. Okay, now I'm not talking about the diploid cell. Okay, I'm talking about the haploid cell. So whatever the haploid cell contains, the genome which is present in the haploid cell nucleus, that amount of the genome is usually represented in picograms. Okay, so that is called as the C-value. Okay, and C-value varies for different organisms. Okay, so different organisms contain different amount of the DNA. And then we have the concept of C-value paradox. Okay, so this tells that there is no correlation between the genome size and the complexity of the organism. Okay, so you should not compare the complexity of the organism with the genome size. Okay, so the different organism have much more DNA when you compare with the body size. Okay, so the body size may be small. Okay, so like Drosophila, but the Drosophila's genome is much bigger than any kind of similar insects. Okay, so that is why that is what this C-value paradox is. Okay, C-value paradox actually tells that there is no consistent correlation between the genome size, which is also called as the C-value, and the complexity of an organism. Okay, if you look at the graph over here, the graph over here, the insected Drosophila has 165 megabase pairs of DNA. Okay, so 165 megabase pairs of the DNA, and the size is like, in this size. Okay, the small dot, what if it just resembles the size of a dot. Okay, and if you compare the DNA of the human cells, okay, whatever the human cells, that DNA present in the human cells, it contains the 1600 megabase pairs. Okay, the cell of the Drosophila is very, very small when you compare to the Homo sapien cell. Okay, and it just 100 times bigger than the, so not 100 times, it actually 10 times bigger than the Drosophila's DNA. Okay, so human cells contains the 10 times greater DNA than the Drosophila's DNA. But when you compare with the body size, Drosophila's body size is much more smaller, like 1000 times smaller than the mammalian body size. Okay, so mammals have a very huge body size when you compare with the Drosophila body size. Okay, so this C-value paradox, it tells that you should not compare the body size with the genome size. Okay, a lot of things were also there, like here we have the fishes. Okay, so it might be the sharks, whales, the big fishes. Okay, and when you compare the genome with in the fishes, okay, so the mammals and the fishes all contains almost same amount of the base pairs. Okay, so but the size of the fishes is quite small, and when you compare to the mammals. Okay, so this is what the C value paradox is, and there is also another value called as the G value. Okay, so it tells about the number of genes that are present in the haploid genome. Okay, so all these things, they only talk about the haploid cells, not about the diploid cells. Okay, and G value represents the number of genes found in the haploid genome, including the known ORFs. Okay, open reading frames. Okay, so this is about the counter length C value paradox on the G value, which actually tells about the genes number of genes present in the blood cell. Okay, now the main concept, that is about the denaturation and the renaturation. Okay, so denaturation is separating the double helical strands of the DNA. Okay, or the double stranded DNA molecule. Okay, so when you break the hydrogen bonds, they separate, and when you disrupt the base stacking interactions, the DNA gets denatured. Okay, so separation and denaturation is carried out in the presence of heat or some kind of formaldehyde solutions. Okay, so when you supply them, heat break down the hydrogen bonds, and formaldehyde, formamide or the urea, they disrupt the base stacking interactions. Okay, so heat also does the denaturation thing, but this process is very abrupt. Okay, abrupt in the sense very slow. So in order to accelerate the process of denaturation, you need to supply some reagents known as urea or formamide. Okay, so these urea and formamide, they tend to release hydroxyl ion. So these negatively charged hydroxyl ion will be able to bind with the DNA bases, and break the base stacking interactions, and the heat will break the hydrogen bonds. Okay, so that the entire DNA strand get denatured. Okay, if you supply only heat, only the hydrogen bonds will be broken down. Okay, and the strands will stay intact. Okay, so there is no disruption of the strands. Okay, the two antiparallel strands. Okay, so when you want DNA strands to be intact, so you should not supply the urea and format. Okay, so when you want to disrupt everything, okay, when you want to break down every single molecule or every single nucleotide of the DNA, so then you can use the heat along with the urea and formaldehyde solutions. Okay, so these chemicals enhance the aqueous solubility of the purine and pyrimidine groups, hence separation of the double strands or the disruption of the double strand will be much easier. Okay, so lot of DNAs, they contain specific regions where the purine rich sequences or pyrimidine rich sequences are more abundant. Okay, like some sort of regions contains the adenine and thymine rich regions. Okay, and some sort of sequences contains the guanine rich regions. Okay, so these sequences which contain guanine rich guanine rich regions. Okay, so these regions tend to have higher melting point. Okay, so they contains the triple bonds between the DNA strands. Okay, and to break the triple bond, okay, we actually require a lot of energy. Okay, so that heat has to supplied in more amount. Okay, so to break the guanine rich regions. Okay, so this is what called as actual denaturations. Okay, and the melting temperature also varies in the DNA. So the region which contains more adenine bases will be having a less TM or less melting temperature. The region which contains the guanine rich sequences, they will be having more TM or the more melting temperatures. Okay, so this is how we separate the DNA strands. Okay, so first what we have is a native DNA. So when you supply the heat, and when you mix urea or formamide to the solution which contains the DNA. Okay, so if you mix the urea and formamide, and then when you heat it, they will remove the hydrogen bonds between the strands, and then the strands get separated. Okay, and when you cool down the temperature, when you decrease the temperature, or when you cool down the solution, okay, so the strands again get renaturated. Okay, renaturation is not again combining the antiparallel strands of the DNA. Okay, so that is what called as a renaturation. Okay, so renature also requires the special conditions. Okay, and the amount of single stranded DNA renatured, that will be explained by the reassociation kinetics. Okay, so the double stranded DNA, it absorbs the wavelength of 260 nanometers, as I've said you in the previous classes. Okay, so this is also known as the UV absorption. okay, and different nucleotides have the different molar excision coefficients, okay, while adenine has the greatest of it, okay. So when you know separate the DNA strands, the absorption of a wavelength, okay, 260 nanometer light, okay, so it increases, okay. So whatever the absorption point you are seeing, okay, or whatever the efficiency of the absorption you are seeing at 260 nanometer 260 nanometer wavelength, you know, when the DNA is in a double stranded form, okay. So this efficiency gets increased, you know, when you separate the DNA strands, okay. So that means, you know, double stranded DNA, assume that, you know, the double stranded DNA has a 5000 molar efficient coefficient, okay, value, okay. When you separate this double stranded DNA into single stranded DNA form, like this ssDNA, okay, so it will have the greater molar excision coefficient, or the greater efficiency of the absorbing those 260 nanometer wavelength light, okay. So this you know kind of increasing the way absorption spectra is regarded as the hyperchromicity or hyperchromic effect, because this effect is also called as the hyperchromic effect, okay. So this is caused due to the exposure of the pyrimidine rings or the purine rings to the light, okay. So when these aromatic bases, you know, they get exposed to the light, you know, they absorb more light, okay. So this absorption of light in more amounts, you know, that will be seen in the case of single stranded DNA forms, okay. So when they are in the double stranded forms, you know, some sort of hindrance will be there, and this hindrance causes the less absorption of the light, okay. So this increased absorption is called as the hyperchromic effect, okay. So that is what I've gave you in third point, okay. So the DNA, I'll read it from here. If a denaturation is allowed spectrophotometrically by monitoring the absorbance of light at 260 nanometers, it is observed that the absorbance at 260 nanometers increases as the DNA become denaturated, okay. So that means, you know, whatever the actual absorbance is there in when you have passed the, you know, light to this double stranded DNA mixture solution, okay. So that absorbance is increased when you separate this when you have separated this double helical DNA into single strands, okay. So this increased absorbance, or the, you know, this phenomena is also called as the hyperchromic effect, or you can also call as the hyperchromicity or hyperchromism, okay. So this is due to unstacking of the bases, okay. So when you unstack the bases, purine bases and the pyrimidine bases, which contain the aromatic rings, so these aromatic rings are much more exposed to the environment and they absorb much more light, okay. So that is what causes the absorbing to increase and resulting in the hyperchromicity effect, okay. So now when you plot a absorbance graph, absorbance at 260 nanometers against the temperature, okay, so that the of a DNA solution, it tells that, you know, there is a little denaturation occurring below the 70 degrees Celsius, okay. So that means, you know, actual denaturation happens above 70 degrees Celsius, not the, you know, not between the 50 degrees or 60 degrees, okay. So there will be a little denaturation happening below 70 degrees Celsius, okay. So this is what the graph where tells you about, okay. So when you plot a graph between this temperature and the absorbance, you know, it will tell you that there is a little denaturation below the 70 degrees Celsius temperature, and, you know, there is a greater amount of the denaturation happening after the 70 degrees Celsius temperature, okay. And when you cool down the temperature of the mixture, then the separated strands get intermixed, okay, like they combine again in anti-parallel direction, okay. So one strand binds with the complementary strand and, you know, forms into the double stranded DNA form, okay. So and they also turn into the double helical forms, okay. So this condition is known as the renaturation, okay. So separated single stranded DNA forms combining to form the double stranded DNA forms, okay. So this is called as the renaturation, okay. And renaturation mainly happens when there is a lower temperature, okay. Okay, so in higher temperatures there will be the denaturation, and when you cool down the temperature, or when the lower temperatures are supplied, then the single stranded will again recombine and renature themselves, okay. Renature is what getting the original form of DNA, okay. So as I've said you, the regions which contain the 80 rich bases, you know, they will have the lower TM values, because, you know, the adenine and thymine, you know, they base pairs with the they get base paired by the 2 hydrogen bonds, while guanine and cytosine, you know, they get base paired by the 3 hydrogen bonds, okay. Because of 3 hydrogen bonds, lot of heat will be required, and the melting temperature also rises, okay. So the melting temperature for this guanine rich regions, or the guanine cytosine content containing DNA sequences, they will have the melting temperature around 90 degrees Celsius. Okay, while the sequencer which contains the adenine and thymine rich bases, okay, so these sequences will have the melting temperature around 70 degrees Celsius. Okay, so this is what melting temperature, this is what the variance of melting temperature is seen between the AD rich sequences and the guanine rich sequences. Okay, so as I've said you, the renature requires a lot of special conditions. Okay, if the special conditions are discussed in the fourth point. If you look at this fourth point, if a heat denatured DNA solution is cooled slowly, which we also called as annealing, which we will be talking in the PCR reaction. Okay, so annealing is what? Combining a primer to the parent DNA strand. Okay, so or annealing also called as a annealing also speaks about the renaturation. Okay, so when heat denatured DNA solution is cooled down slowly, and annealing happens and hold the solution about 25 degrees Celsius, which is actually a lower temperature below Tm and above a concentration of 0.4 molar Na plus for several hours, some amount of the DNA or that is for 50 to 60% is renatured. Okay, rapid cooling does not reverse the denature, but if the cooled solution is again heated, then cooled slowly renaturation takes place. Okay, so rapid cooling in the sense, just taking the solution mixture from the hot water bath and keeping this mixture of solution mixture into the freezer. Okay, so that is what called as a rapid freezing, or we also call them as the heat shock treatment. Okay, so this kind of shock treatment or rapid cooling doesn't allow the normal single strands to get paired. Okay, complementary pairing with the another single strand. Okay, so this won't be happening. Okay, so the renature thing won't be happening. The renature will only happens when you cool down the temperature very slowly and allow the single strand DNAs to search for their complementary strands. Okay, and giving some time to get hydrogen bonded. Okay, so giving some time to get bonded with the another single stranded DNA. Okay, so this kind of slow cooling will only give you the complete renatured DNA. Okay, so this is what basic concept about the denaturation and the renaturation. Okay, so when you denature the DNA, the density increases, the buoyant density increases, hyperchromicity will be seen. That means the absorbance of the 260 nanometers also increases, the effect increases. Okay, a lot of things happens. Okay, the exposure of the pyrimidine bases to the environment also increases. Okay, so this is involved in the denaturing process. Okay, so when you do this renaturation, when you cool down the temperature and allow the single strand to mix, okay, the absorbance will get decreased, the density will get decreased, bond density will get decreased. Okay, so the denaturation and renaturation will have the almost opposite points. Okay, or concepts. Okay, so this is about the denaturation, the renaturation. Okay, so the kinetics which are involved in the reassociation. Okay, so that is analyzed by the COT values. Okay, COT analysis. Okay, COT analysis speaks about the how much amount of repetitive DNA is there in your nuclear genome, or how complex is your nuclear genome. Okay, so that concept will be taught by this COT analysis. Okay, COT analysis says that how your genome is complex, or how much repetitive sequence are there in your genome. Okay, so this is known as the analyzing these kind of things is known as the COT analysis. Okay, so and it is based on the principles of DNA reassociation kinetics. That means when you are cooling down the temperature, the single strand getting absorbed. Okay, so how much time it is taking and how much DNA is getting formed. Okay, the renatured DNA is formed. Okay, so that is called as reassociation kinetics. Okay, so this analysis involves the DNA reassociation kinetics and measures how quickly how fastly the single stranded DNA sequences find their complementary strands and reform the double stranded DNA strands. Okay, double stranded DNA. Okay, and this renaturation of the DNA, it depends on the random collision. That means one set of single stranded DNA sequences randomly collides with the another set of single stranded DNA sequences. Okay, so it collides randomly, and when it finds a complementary strand, then it goes and binds with the complementary strand. Okay, so that is what random collision is tell you. And it follows the second order kinetics. Okay, that means the greater the substrate concentration or the single stranded DNA concentration, the greater will be the renaturation process. Okay, so as the amount of single stranded DNA is getting decreased in the solution, the rate of reassociation also get decreased. Okay, so then that is what the second order kinetics says about this reassociation kinetics. Okay, and this technique or this COT analysis was first developed or first observed by the Roy Britton and his colleagues in the Carnegie Institution of Washington. Okay, so in 1960s, they have analyzed they have analyzed the repetitive sequences which are present in the nuclear genome, and they have analyzed how complex the DNA or how complex the genome was. Okay, so how do you perform this COT analysis? Because there is a simple procedure which we do during the renaturation process also. First, you need a hydroxyapatite column. So when you perform a column chromatography, so you will be seeing this hydroxyapatite column thing. Okay, so first what you do is you supply the DNA mixture, the denatured or shared DNA mixture into the hydroxyapatite column. Okay, and then you pass on some sodium phosphate buffer, a little concentration of the sodium phosphate buffer or low concentrations of the sodium phosphate buffer to the hydroxyapatite column. Okay, so this sodium phosphate buffer first elutes the single stranded DNA molecules, and then you have to wash the column with the higher concentrations of the sodium phosphate buffers. Okay, so this higher concentration now elutes the double stranded DNA forms. Okay, so when you wash the hydroxyapatite column initially with the lower concentration of the sodium phosphate buffer, okay, so you extract the mixture, okay, which contain the single stranded DNA forms, and second time also you have to do the same thing to extract the mixture when you wash this hydroxyapatite column with the higher concentration of the sodium phosphate buffer. Okay, so this two different mixture will tell you how much DNA is renatured and what's the time for renaturation process. Okay, what's the time required for the single stranded DNAs to find the complementary strand and get hydrogen bonded with the complementary strands. Okay, so that will tell you this mixture analysis will tell you the reassociation kinetics. Okay, so I've gave you in this column in this point the amount of single stranded and double stranded DNA is measured by rapidly diluting the sample. Okay, how? Which slows the reassociations. Okay, and then binding the DNA to the hydroxyapatite column. Okay, first you have to share the DNA, denature the DNA, and then pass the mixture to the hydroxyapatite column. Okay, so this will the column chromatography, and this column is first washed with the low concentrations of the sodium phosphate buffer. Okay, why is low concentration of the sodium phosphate buffer? Because they have a lower concentration will elute the single stranded DNA molecules. Okay, understood? Which elute the single stranded DNA, and then high concentrations of the phosphate buffer is used to elute the double stranded DNA molecules. Okay, so when you are washing with the lower concentrations, at that time you have to extract the mixture, okay, or you collect the mixture, and then for the second time when you are using higher concentration of the buffer, at that time also you have to collect the mixture. Okay, so the difference between these two things, okay, the how much single stranded DNA has formed into double stranded form, or how much single stranded DNA is left behind. Okay, so that is analyzed by the spectrophotometer by passing the wavelength of 260 nanometers light of 260 nanometer wavelength. Okay, so this difference will give you the analysis. Okay, so if the DNA is in a repetitive nature, or if the DNA is unique nature, or contains unique sequences. Okay, so this kind of observation will be made by the spectrophotometric analysis. Okay, so there is an equation when DNA is shared into uniform fragments about 300 base pairs length. Okay, so uniformly and allowed to renature, then the rate of renature of DNA of denatured DNA is expressed in the terms of dC by dt is equals to minus k c square. Okay, so dC or the capital C represents the concentration of the single stranded DNA at time 0. Okay, so initial time, and minus k represents the second order rate constant. Okay, remember that concentration of the single stranded DNA always inversely proportional to the second order rate constant, that is k. Okay, so the second order rate for two complementary strands coming together is given by the rate of decrease in C. Okay, and starting with the concentration C is 0 or C naught of a completely denatured DNA at time 0, the amount of single stranded DNA remaining at the at that time is given by the equation C by C naught is equals to 1 by 1 plus K into C naught into T. So this equation will give you the starting concentration of completely denatured DNA. Okay, how much the single stranded DNA is present in initially in the mixture. Okay, so that is given by the second equation. Okay, and after reassociating 50, when the DNA got 50% reassociated, okay, you have denatured it and you have allowed it for the renaturation. Okay, now the 50% of the DNA has got reassociated. Okay, now the equation will be modified to C naught into T half is equals to 1 by K. I remember, as the concentration of the single stranded DNA increases or the C naught value increases, okay, lower will be the reaction. Okay, that means, as I said you, it follows the second order rate of concentration. So second order rate constant. Okay, second order rate kinetics. Substrate constant, this rate of reaction, it will be majorly dependent on the substrate concentration. Okay, as the substrate concentration decreases, as the C naught value decreases, the rate of reaction increases. Okay, so that means, when there is a higher C naught value, the reaction rate will be slower. Okay, so that means, when there is a C naught value or the concentration of this single stranded DNA, when it is higher amount, okay, so that the rate of the reaction rate will be very low or very slow. Okay, so that is given by the this formula, and it will tell you about the reassociation kinetics. Okay, so based on this reassociation kinetics, these scientists, okay, Roy and Bitter, because they found that there are three types of copies or three types of DNA sequences are present in our nuclear genome. Okay, so one is the highly repetitive, one is the moderately repetitive, and one is the single copy repetitive DNA. Okay, or you can also call that as a non-copy repetitive, because lot of unique sequences are present in the single copy DNA. Okay, so these were the major findings based on the reassociation kinetics. Okay, so first one is the highly repetitive DNA. So it occupies about 20% of the genome. Okay, and it has very low COT values. Okay, that means 10 power minus 4 to 10 power minus 1. Okay, so when you plot a concentration of single stranded DNA, okay, so with the log COT value, so you will get a graph. Okay, so initial stages will indicate the highly repetitive components. Okay, and the mid stages represent them moderately repetitive components. Okay, and the higher COT values represent the single component or the unique sequences of the DNA. Okay, so this is what the graph actually says. Okay, and then what we have is the highly repetitive one. Okay, so these have the low COT values. Okay, that is in the range of 10 power minus 4 to 10 power minus 1. Okay, and most of the satellite DNA, like mini satellites and the micro satellites, okay, so they will be having lot of repetitive sequences like adenine and thymine, sequences which are repeated rapidly. Okay, so this kind of repetition will make the DNA sequences to be highly repetitive. Okay, so this will make the DNA to have the low COT values. Okay, and the examples include the satellite DNA, mini satellites, and the micro satellites. Okay, next comes the moderately repetitive DNA, and this occupies about 40% of the genome. Okay, and most of the moderately repetitive DNA sequences will be the transposons. Okay, that is the DNA transposons. Okay, and also we have this lines and signs. Okay, so long interspersed nuclear elements and short interspersed nuclear elements. Okay, so these are the major moderately repetitive DNA sequences which are present in our genome. Okay, and they occupy about 40%. Okay, and they have the intermediate COT values. That means the renaturation will be minimal. Okay, so minimal, or in the renaturation occurs normally. Okay, so these sequences they are interspersed throughout the genome. Okay, so that means on entire chromosomes, these moderately repeated DNA sequences are present. Okay, so they are interspersed. Okay, they are dispersed throughout the chromosomes. Okay, and then we have the single copy or non-repetitive DNA sequences which occupies about 30% of the genome, and mostly the protein coding enzymes. Okay, they fall into this category. Okay, so these sequences typically include most of the genes that code for the proteins. Okay, and they also have the higher COT values. That means they reassociate very slowly. Okay, I've said you right, the rate of reassociation decreases as the COT value increases. Okay, whenever there is a single stranded DNA concentration at higher values. Okay, so at that time the reassociation kinetics will be very slow. Okay, so that means these single copy sequences or non-competitive DNA sequences reassociate very slowly at high chord values. Okay, so that their chord values will be very large. So that is around the 10 power 3 to 10 power 5 in this range. Okay, now as I've said you, most of the genes which code for the proteins, they fall into this category. Okay, so either they might be the protein coding genes. Okay, and we also have the RNA coding genes. Okay, like ribosomal RNA and messenger RNA. They fall into the motility repetitive component because these genes are also clustered throughout the chromosomes. Okay, so they fall into the motility repetitive. They are not the singular low copy repeated sequences. Okay, only the protein coding genes which are expressed minimally, they fall into this non-repetitive sequences or single or low copy repetitive sequences category. Okay, so this is what about the reassociation kinetics. Okay, and basic terms like C value and C value paradox. Okay, and in the next class we'll be discussing about the kinetic classes of the DNA, so which includes the interspersed elements and tandem repeats and DNA transposons. Okay.